Cloud. Okay. So we ended here last time. So part two. So cells have an optimal pH. Okay. So I'm sure you guys heard the term pH before. I'm not sure you guys understand what it really means. So pH is a scale based on the amount of hydrogen ions acid in the solution. Okay. Both chemical reactions in the cells occur around pH 7, more specifically 7.4. Okay, and that is a very narrow range. Okay, like you cannot get too high or too low, because if you get too high or too low, you get into acidosis and stuff, and that's really bad. Okay, okay, so acidic solutions have a low pH and therefore a high concentration. pH is the opposite of so if you have high pH, that means you have low H plus levels. If you have low pH, it means you have high H plus levels. Okay. And the way we control our pH is something called buffer. Buffer solutions help maintain a constant pH by absorbing or releasing hydrogen atoms into the solution. So if we encounter more acid than we want, the buffer absorbs some of the acid. When we need acid, the buffer releases some of the acid, and that's what keeps us safe. Okay. Okay. The next section really is a quick primer on organic chemistry. Organic molecules, by definition, have to have carbon and hydrogen. Okay. Methane is a simple organic, the most simple organic molecule. It doesn't get any simpler than that. 1 carbon, 4 hydrogens. Okay. Okay. We'll skip the functional groups. Okay. Organic molecules are biologically important. Okay, so there are 4 main types of biological molecules. Okay. So let's talk about what organic molecules are made out of. Okay, organic molecules are usually made out of something called a monomer. Mono means 1. Okay, so and mer actually means unit. So monomer, single unit. Polymer, multiple units. So we take monomers and we string them together into a polymer. Okay, and we do this with carbohydrates, we do this with proteins, and we do this with nucleic acids. Okay. Okay, the process by which we do this is called dehydration synthesis. Dehydration synthesis is the type of chemical reaction in which we take 2 monomers and attach them together to form a dimer, and from dimer eventually we join them together to form a polymer. Okay, so the whole key to this, okay, is water. Okay, as part of the reaction, a water molecule is released. So you can see H2O right here. We're going to cut that out. So dehydration, removal, remove, hydration, water, synthesis, to put 2 things together. We remove water and we put 2 things together. Okay. So dehydration synthesis takes 2 units and joins them together. The opposite of dehydration synthesis is something called hydrolysis, okay, or hydrolysis. Okay. So hydrolysis breaks them apart. So hydro is the use of water, lysis break apart. So I use water, they say I do the opposite. I attach OH to one side, so this side will be the OH, and then this side will be the H, and then I'm back to where I was before. Okay. Yeah. Okay, so one of the 4 types of biologically relevant molecules. There's something called carbohydrate. Carbohydrate, okay, is exactly what it sounds like. Carbohydrates have carbon and water. Okay? So their formula will be CH2O to the nth power. And so like this is C5H10O5, C6H12O6. So CH2O to the nth power. Okay. Okay. And then clear all this first. Okay. So saccharide is the term for sugar. So monosaccharides are 1 units of sugars. So ribose, glucose, fructose are single unit sugars. I take single unit sugars and I combine them to form 2 unit sugars. 2 unit sugars disaccharide. So many unit sugars, polysaccharides. Okay, so single unit and 2 unit sugars we usually use for quick energy. Okay, polysaccharides are very long chains of carbohydrates, and we either use them for structure, so cellulose. Chitin is for structure, or energy, like long medium-term energy storage. So not quite as energy storage as fat, but more efficient than sugar. So glycogen and starch. Glycogen is used in animals, starch is used in plants. Okay. Next. Proteins have many different functions, and you can see all the functions of proteins, and we'll talk more about that later. Proteins are made out of these things called amino acids. Okay, there's 20 different ones. Okay. The only thing different between each amino acid is this R group. And some amino acids have small R groups, some of them have large R groups, some of them are polar, some of them are non-polar. It's just really complicated, but we'll keep it simple because the different R groups tell you different things about the molecule. Okay. Again, same thing. Proteins can be synthesized and broken down. You make them through dehydration synthesis. You break them down through hydrolysis. Okay. 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 So we'll come back to this slide in a second. Okay. Okay. There are 4 levels of structures in protein: primary, secondary, tertiary, quaternary. Okay. The primary sequence or primary structure is also known as the amino acid sequence. These are how the protein is actually strung together into a string. Okay, so the amino acid sequence is right here. When you string this together, they form these secondary structures, alpha helixes or beta sheets. The secondary structures will form tertiary structures, and then tertiary structures will form quaternary structures. Okay, so there's many levels of folding. Okay, and everything has to go right for the protein to be functional. Okay, if I take this structure right here and I unfold it, unravel it, okay, I can screw it up. Okay, so if I take the structure right here, you see that the beta sheets, sorry, the beta sheets are here. So the beta sheets are here, right? And then the alpha helixes are here. You see one there, you see one there. Okay. If I take this structure and it forms structure, if I heat it up and mess it up, it changes the conformation from white gluey stuff to, or clear gluey stuff to white, not so gluey hard stuff. And so it no longer works. Okay. Okay. Nucleic acids carry genetic information. Okay. So nucleic acids are either made out of DNA or RNA. Okay. The monomers of nucleic acids are nucleotides. There's always 3 parts to the nucleotide. Okay. A nitrogenous base. Okay. A sugar and a phosphate group. Okay. Nitrogenous bases are found in different nucleotides. Okay. All DNA and RNA have A, C's, and G's, always. DNA also has T's. Okay? RNA does not have T's, RNA has U's. We'll learn much more about the rules of DNA and RNA in a later chapter, but just introduce the concept. Okay. And again, dehydration, synthesis, and hydrolysis. Okay? All right, so the last group we'll talk about are fats. Okay, the other 3 groups that we talked about function primarily in water. Fats do not. Fats or lipids are hydrophobic molecules. Okay, so they stay away from water as much as possible. Okay, fats are also the only group of these things that are not made out of monomers. Okay, fats are made out of 2 units. They're made out this part right here, which is called glycerol, and long-term fatty acids or long-chain fatty acids. Okay. So this is a triglyceride, 3. This is glycerol with things attached to it, so glyceride. So 3 things attached to it, so triglyceride. Okay, this is actually, triglycerides are actually long-term energy storage. Okay. All right. Fatty acids are either saturated or unsaturated. Saturated fatty acids have no double bonds, and therefore they are straight. And because they're straight, they pack together very tightly. And because they pack together very tightly, they form solids. Okay, unsaturated fats, okay, have at least 1 double bond, and because the double bond throws this out of shape, it's no longer straight, and because they're no longer straight, they can't fit together very tightly, and they're usually oils. Okay, trans fats are really kind of weird. They have a double bond, but they are straight, and our body doesn't actually know how to deal with this. So trans fats, our body doesn't know how to break it down, so they stay around in our bodies for a much longer period of time than they should, and that's what makes them bad. So that's why you see they purposely try to take trans fat out of foods now. Okay. The second class of lipids are a small class, but really important class. They're cholesterols. Cholesterols are a member of steroids, and they're the cholesterol that we use in our blood can get turned into other things like testosterone for males and with estrogen and progesterone for females, and they actually do a lot of like control a lot of our behaviors. Okay. All right. Hopefully that explains everything. And I will see you guys in class. And